Let's talk about the chemical nature of DNA. Back in 1869, a German chemist named Friedrich Miescher made an important discovery. He extracted a white substance from the nuclei of human cells and from fish sperm. Since this substance was always associated with the nucleus, he named it nuclein. Later, scientists realized that nuclein was acidic, so it became known as nucleic acid. At first, biologists did not pay much attention to it because they did not know what role it played to the cell. But in the 1920s, a biochemist named P.A. Levene worked out its basic structure. Levene found that DNA is made up of three main components: a phosphate group, a 5-carbon sugar known as pentose sugar, and nitrogen-containing bases. These bases are of two types, purines and pyrimidines. Purines, adenine A and guanine G, pyrimidines, thymine T and cytosine C. And in case of RNA, instead of thymine, we find uracil U. Levene also discovered that these components form repeating units called nucleotides. Now, let's see how a nucleotide is arranged. The nitrogen base attaches to the carbon number 1 of the sugar. The phosphate group attaches to the carbon number 5 of the sugar, and there is also a hydroxyl group OH at carbon number 3. This arrangement is very important. Why? Because the phosphate group on carbon number 5 of one nucleotide can chemically react with the hydroxyl group on carbon number 3 of another nucleotide. This reaction releases a water molecule, a process known as dehydration synthesis. The result is a strong covalent bond between the two nucleotides known as phosphodiester bond. In this bond, the phosphate group connects two sugar molecules through oxygen atoms. Because of this bonding, nucleotides can join together again and again, forming long chains, thousands of nucleotides linked in a row. An important thing to remember is every DNA or RNA strand always has a free phosphate group at the 5' end and a free hydroxyl group at the 3' end. This polarity or direction is what allows DNA or RNA to build linear chains that carry genetic information.